name's Eva Booth and I'm a final year forensic investigation student at Staffordshire University. This presentation will cover my dissertation where I worked alongside the Zoological Society of London in research into the subject of how confidently can we detect that wildlife have been shot with air weapons or 2.2 long rifles. There is little previous forensic research into determining whether animals have been shot, which makes identifying wildlife crime difficult. The aims of my project are to look at differences between feathers shot with an air weapon compared to those shot with a 2.2 long rifle, to look at differences between lead and lead-free ammunition in terms of damage and elements present on the feather, to use a scanning electron microscope with energy dispersive X-ray analysis to measure metal content left on shot feathers compared to control samples, and to analyse unshot feathers compared with shot feathers and look if environmental factors could affect metal content on the feathers. Pigeon were taken from the population shot for crop protection. The tail feathers were plucked from these pigeons and 5 feathers were shot for each type of ammunition. The rifles that would be used were a Logan Solo FX air rifle with lead-free air rifle pellets from Field Target Trophy Green and Crossman Accupel lead pellets. A Seiko 2.2 long rifle with lead Ely bullets were used. Both rifles were cleaned prior to this stage to reduce contamination of metals through the different types of ammunition. The feathers were first analysed using a Nikon low-powered dissecting microscope. This was to check for any visible elements of metal on them and also to identify what the damage looked like on a close-up scale. From the images taken with the microscope, it was clear that no traces of metal could be seen at this low magnification, but there were some patterns in the damage from the shot which occurred on the feathers. When the site of entry of the bullet or pellet was on the pigeon feathers was on the barbs coming off the shaft, the barbs seemed to curve outwards and then back in around the site of entry, which can be seen in the top photo, whereas on the kite feathers it would break off in a straight line, which can be seen on the bottom photo. The picture on the left shows how when the site of entry was on the area of the barb closest to the shaft, the barbs would be ripped off. On the picture on the right, it shows how when the site of entry was on the shaft of the feather, the fracture or break on the shaft would be jagged. The 2.2 rimfire was more destructive than the air rifle on both pigeon and kite feathers, resulting in a larger area of damage. This could be due to the muzzle velocity and energy, which is the energy that the bullet has as it leaves the barrel. Rimfires have higher energy and muzzle velocity than air rifles. The Ely lead bullets have a much larger muzzle energy of 129.79 joules compared to the lead air rifles, which have a muzzle energy of 15.18 joules, and the lead-free air rifle pellets, which have a muzzle energy of 15.78 joules. The scanning electron microscope was used to analyse the shot feathers for each ammunition and an unshot feather from each bird to act as a control feather. This would act as an indicator of any elements present on the feather before they were shot. Images were taken of any areas of interest. The unshot feathers appeared grey in colour with no large areas of heavy elements. There was only one particle by itself on an unshot feather which appeared to be sitting on the surface of the feather. On pigeon 1 there was a particle visible measuring 110 micrometers by 50 micrometers. On the unshot feather from pigeon 2, the particle visible when analysed measured 9.7 micrometers by 14.5 micrometers. And the unshot feather from pigeon 3 had a particle on measuring 93.5 micrometers by 40.3 micrometers. On the 2.2 rimfire lead shot feathers, there was a large quantity of bright areas on the feathers, indicating lots of areas of heavy elemental transfer. These areas appeared as smear-like patterns along the surface of the barbs and the barbules on the feather. The areas of heavy elements were located around or below the site of impact and were dense in these areas. There was evidence of loose particles of heavy elements on the surface above the shot site, which were spherical in shape and had a rough appearance on their surface. On the lead air rifle pellet shot feathers, there appeared to be smears of heavy elements stretching across the barbs. There were various particles sitting on the surface measuring between 3 micrometers and 7 micrometers. With the feathers shot with the lead air rifle pellets, there was little evidence of areas of heavy elements around the shot site or on the feather in general. The general pattern that there was that there was some small smears along the barbs and the shaft of the feather, and a minimal number of spherical particles sitting on the surface of the feather around the shot site. On the feathers shot with the lead-free air rifle pellets, there is very small areas of heavy elements on the feather around the estimated site of entry on the shafts of the barbs and on the ends of the barbules. These particles are less concentrated and are more spread out with spherical shape and a rough appearance. The particles appear to be sitting on the surface of the feather. There is one small smear on one of the feathers at the site of impact, which can be seen on the photo on the right. The unshot kite feathers had areas of white all over, but it was more compact in between the barbules, with some smears or build-up of particles on the barbs. There are brighter areas at the end of the barbs where it joins the shaft, and they look as though they are spread over the surface of the outer area. These were not as bright as other areas of heavy elements and were more faded. This is shown in the top image on the right. The kite feathers that have been shot with the 2.2 rimfire lead bullet have bright areas around the site of impact, on the shaft and on the ends of the barbs that have been snapped off by the bullet, and on the tips of the barbules. The pattern created was a smear-like pattern similar to that on the pigeon feathers shot with a 2.2 bullet. The kite feather appeared similar to the pigeon feather shot with the same ammunition, but with slightly less areas of heavy elements on. The air rifle lead ammunition shot feathers had small areas of heavy elements smeared along the barbs and on the tips of the barbules around the site of impact. There were only small amounts of particles sitting on the surface. The elemental transfer as a result of the shooting seemed to be mainly on the site of impact of the ammunition or around this area. This could be a problem if the exact site of impact is not known on the bird, as there may be no elemental transfer on any other parts of the feather. This could result in the bird's death being misrecorded, and it would not look like it had been shot. The elemental transfer on the feathers shot with both types of lead ammunition appeared as smears along the barbs, and there was an occasional particle on the shot feathers. The majority of these particles were less than 10 micrometers in size, which is consistent with findings from a study carried out by Wallace et al. In 1984, where it was found that particles produced by ammunition are found to be between 1 and 12 micrometers in size, with spherical shape and an irregular surface texture. All particles found on the unshot pigeon feathers were larger than 12 micrometers in size. The morphological form of particles found on the unshot feathers were an elongated oval shape at around 50 micrometers in width and nearly 2 times this in length. This indicates that there is a difference in particles found on shot feathers as they are a result of gunshot residue compared to the larger particles on unshot feathers. Elemental analysis was carried out using energy dispersive X-ray analysis. Here are the 4 charts showing the differences in elements between ammunition types and unshot feathers. In general, carbon, nitrogen and oxygen were present amongst all the feathers in high levels. The elements that appeared on the shot feathers that were absent on the unshot feathers were chlorine, barium and tin. There was lead present on the lead shot ammunition, but there was one particle of lead found on the feather taken from pigeon 2. When the EDX analysis was carried out on this unshot feather, the spectrum results showed that 51.8% of the weight of the area analysed was lead. This particle was a single particle and the only one present on the feather. From the appearance of this particle, it's clear that it's sitting on the surface and does not look the same as the particles which contain lead on the shot feathers. This is most likely due to contamination and has been picked up from surrounding areas. Here are the 4 charts showing the difference in elements between the kite feathers. In general, there is high levels of carbon, oxygen and nitrogen on all of the shot and unshot feathers. The metals tin, lead and barium were present on some of the shot feathers, but absent on the unshot feathers. Tin was present on the lead air pellet shot feathers in low levels and on the lead-free air pellet shot feathers at higher levels due to the pellet being made of a tin alloy as an alternative to lead. ImageJ is a software used to analyse the percentage of white area out of a total image. This was used on the scanning electron microscope images to analyse the white areas, which are areas of heavy elements. The shot feathers were compared with the unshot feathers, and it was found that the 2.2 rimfire lead bullet had the greatest difference in white area between the shot and unshot feathers, with 239.9 times as much white area on the shot as the unshot feather. The lead air rifle pellet was next, with 3.2 times as much white area on the shot feather compared to the unshot feather, and the lead-free air rifle pellet had the least difference in white area, with only 1.2 times as much white area on the shot feather compared to the unshot feather. This study has found that while environmental factors can produce elements similar to those found in gunshot residue, energy dispersive X-ray analysis can identify elements which would not normally be present on unshot feathers such as barium and lead. By using the scanning electron microscope to analyse the morphological characteristics of particles transferred from gunshot residue onto the feather, along with ImageJ to analyse the percentage of white areas on those images, it is clear that there is a difference in appearance and quantity between environmental deposition of particles and particles from gunshot residue or ammunition transfer. There are larger areas of heavy elements on the shot feathers which often appear as smears or small particles compared to the unshot feathers which were larger surface particles. So to conclude, it is possible to determine if wildlife such as birds have been shot by analysing the feather using scanning electron microscope with energy dispersive X-ray analysis for elemental analysis and the morphological characteristics of the areas of heavy elements. Thank you for listening.